Hello everyone. Today we shall discuss about the arachidonic acid metabolism which will include the Cox-Lox pathway. So let's begin. Eicosanoids is derived from the Greek word eikosa which means 20. Eicosanoids are lipid-based signaling molecules that play a unique role in innate immune responses. They are derived from either omega-3 or omega-6 fatty acids. Eicosanoids are oxygenated derivatives of three different PUFAs. Eicosapentanoic acid, which is abbreviated as EPA, it's a 20 carbon chain omega 3 polyunsaturated fatty acid having 5 double bonds. Arachidonic acid, abbreviated as AA, is a 20 carbon omega 6 fatty acid having 4 double bonds, so it is also a PUFA. And the third one is dihomogamma-linolenic acid, which is abbreviated as DGLA, and it is a 18 carbon omega 3 polyunsaturated fatty acid having 3 double bonds. So let us look at the classification. Eicosanoids are classified into two main groups: prostanoids, leukotrienes, and lipoxenes. Prostanoids are further subclassified into three groups, that is prostaglandins, prostacyclins, and thromboxanes. So let us look at the chemical structure of the prostanoids. Prostanoids are prostaglandins that are derived from 20 carbon cyclic saturated fatty acid known as prostanoic acid. They have a 5 carbon ring in which is saturated. So this is the general chemical structure of all prostanoids. You can see that there are 20 carbons. The first carbon always has carboxyl group. There is a hydroxy group at the 15th position, a double bond between 13 and 14 carbons, and there is a ring structure where the ring closes between carbon 8 and 12. At the carbons 9 and 11, there are different substituent groups which will vary according to the derived prostanoid acid. The classification of prostaglandins. There are 9 classes of prostaglandins: PGA, B, C, D, E, F, G, H, and I. They are classified into 3 series. Series 1, double bond, precursor is D-GLA or linolenic acid. Example is PGE1. Series 2, two double bonds, precursor is arachidonic acid. Example PGE2. Series 3, there are 3 double bonds, precursor is linolenic acid, and there are 3 double bonds. So you can see that the series indicates the number of double bonds. So wherever there is PGE1, that means it has single double bond. PGE2 means it has 2 double bonds, and PGE3 means it has 3 double bonds. So let us see the biosynthesis of prostaglandins. The prostaglandins undergo biosynthesis. The eicosanoid biosynthesis begins when a cell is activated by mechanical trauma, cytokines, growth factors, or other stimuli. So there is a stimulus that is needed for the biosynthesis of prostaglandin. This triggers the release of the enzyme phospholipase at the cell membrane. The phospholipase travels to the nuclear membrane, and there the phospholipase catalyzes hydrolysis of phospholipid or diacylglycerol by different phospholipases. So in case of diacylglycerol, it is phospholipase C. There are This frees the 20 carbon fatty acids from the membrane. Now let us look at this. So this is a phospholipid. and there are different phospholipases. We will see that all the phospholipases have different sites of action, and that is how they are named. So phospholipase A1 acts at this position, carbon 1 position, and the product formed is lysophospholipid plus free fatty acid. Phospholipase A2 acts at the second carbon position, and it also liberates lysophospholipid plus free fatty acid, and generally the free fatty acid liberated here is a polyunsaturated fatty acid, example arachidonic acid. The third phospholipase is phospholipase C. It always acts at the third carbon, and the product is diacylglycerol plus inositol triphosphate. Whereas the fourth phospholipase, that is the phospholipase D, acts at the phosphate group, and the product is a phosphatidic acid. So these are the actions of different phospholipases. Now the X here at the third carbon where the phospholipase D acts are different lipids that they include choline, ethanolamine, serine, inositol and glycerol. So based on the action of phospholipases, there is a interesting fact that the snake venom has high concentration of phospholipase A2, and when the snake bites, this phospholipase A2 is released into the human or the animal blood, and its action is to release lysophospholipid and platelet activating factor and various prostaglandins, thromboxanes and leukotrienes which cause inflammation, edema. They cause myotoxicity, cardiotoxicity, anticoagulant effects, platelet aggregation, initiation, convulsive effects and edema. So these are the various effects of snake venom depending upon the species of the snake. So it can be it can cause hemorrhage, cytotoxicity, neurotoxicity and myotoxicity. So this is one example of the enzyme phospholipase A2 which is present in the snake venom. Now in our body, the phospholipase A2 acts on the cell membrane, and the product is lysoplatelet activating factor and arachidonic acid. This arachidonic acid undergoes two pathways, the COX pathway and the LOX pathway, based on two different enzymes, cyclooxygenase and lipoxygenase. So the phospholipase A2, which is the first enzyme of the pathway, is inhibited by steroids, and the cyclooxygenase is inhibited by a group of drug known as NSAIDs, that is non-steroidal anti-inflammatory drugs, which we will look at later on. So arachidonic acid undergoes two pathways, COX and LOX pathway. So when it undergoes COX pathway, it produces prostaglandins, and when it undergoes LOX pathway, it produces leukotrienes. Now we come to the phospholipid lecithin, which is converted to arachidonic acid with the help of phospholipase enzyme, and it gives rise to prostaglandin G2. So this is the first prostaglandin to be formed from arachidonic acid. Now this prostaglandin G2 is converted to PGH2, and this PGH2 will give rise to various other prostaglandins and thromboxanes. So prostaglandin D2, prostaglandin E2, prostaglandin F2 alpha, prostaglandin I2 and thromboxane A2 are the products that are derived from the cyclooxygenase pathway. The first prostaglandin to be formed is PGG2, and it gets converted to PGH2, and by various enzymatic reactions, PGH2 is converted to the other prostaglandins and prostacyclins. Only 5 prostaglandins are widely distributed in our body. The Cox-Lox pathway. So let us look at the Cox-Lox pathway in detail. So now let us look at the Cox pathway in detail. So membrane phospholipid under the action of phospholipase A2, membrane phospholipid lecithin will first release arachidonic acid, and this arachidonic acid can also be derived from phosphatidylinositol or from linoleic acid, which is obtained in the diet. So these are the sources of arachidonic acid, that is the membrane phospholipid lecithin, phosphatidylinositol and linoleic acid. This phospholipase A2 is stimulated by or activated by hormones epinephrine, thrombin, angiotensin 2, bradykinin and vasopressin. And it is inhibited by steroids, which we saw in the previous slide. So if the prostaglandin production has to be stopped at this level, steroids may be given. Now, arachidonic acid further undergoes the cyclooxygenase pathway to produce PGG2. PGG2 is now going to be converted to various other products, but arachidonic acid can be converted to leukotrienes by the lipoxygenase enzyme. So let us look at the COX pathway. Now PGG2 is converted to PGH2, and PGH2 will be further converted by different enzymes to other prostaglandins and prostacyclins. So the first product is thromboxane A2, which is formed by thromboxane synthase. The enzyme prostacyclin synthase gives rise to prostacyclin PGI2, and the thromboxane A2 has two main effects. So that is bronchoconstriction and platelet aggregation, thrombosis and vasoconstriction. So how do we remember this? As you can see that the word TXA2 is here. So A stands for aggregation and X you can link with constriction. So it causes constriction in the lungs, bronchoconstriction, and in the blood vessels it causes vasoconstriction. And with A it stands for aggregation, so it causes platelet aggregation and thrombosis. PGI2 has the opposing effects of thromboxane A2. I stands for inhibition, so it inhibits all the actions of thromboxane A2. So let us look at the different effects. So PGI2 is also known as prostacyclin. It keeps the blood circulating, so you can remember that it keeps the blood cycling, and it is acting opposite to thromboxane A2, so it causes vasodilation and it inhibits platelet aggregation. Now we come to the next prostaglandin, that is PGD2. D stands for dilatation, so it causes dilation and it's anti-inflammatory. PGE2 is also derived from PGH2. So as you can see, PGH2 is the central molecule from which all prostaglandins are and thromboxanes are derived. Now PGE2, here E is enlarged, so it stands for all the E stands for fever, inflammation, pain and tumor growth. And PGE2 can be converted to PGF2 alpha, and here F stands for fertility. So PGF2 alpha is used in the contraction of uterine, myometrium, induction of labor. As a medicine, it is used for termination of pregnancy. It also causes bronchoconstriction and platelet aggregation. So it has all these effects. Now we go to the LOX pathway. So the leukotrienes is first converted to LTB4, and from here uh, this LTB4 is causing chemotaxis, and it generates lot of neutrophils. It is quickly converted to LTC4, LTD4, and LTE4, and the 4 here represents the number of double bonds. So all these C, D, and E4 are causing bronchoconstriction. So this is the overall Cox-Lox pathway. Now, the mechanism of action. So, how do these prostaglandins and thromboxanes act once they are released? So, they act by uh, having a effect on adenylyl cyclase. So, we can see here that F2, E2, and D2 prostaglandins, they will activate adenyl cyclase enzyme, which will convert ATP to cyclic AMP, and all the biological effects will be produced. So this is how they act. Now, the function of this prostaglandin is through G protein coupled receptors, and in most tissues, prostaglandin E increases the cyclic AMP level, whereas only in adipose tissue and renal tubules it decreases the cyclic AMP level. And the prostaglandin I activates adenylyl cyclase, and thromboxane A inhibits it. So these are the different effects of different prostaglandins on adenylyl cyclase enzyme. The classification of prostaglandins can also be based on the fatty acid from or the PUFA from which they are produced. So they are good eicosanoids and bad eicosanoids. The good eicosanoids are produced from omega 3 fatty acid because they prevent blood clot by platelet aggregation. They cause dilation of blood vessels, they reduce pain, they decrease cell division, they enhance immune system, and they improve brain function. So they have lot of good effects on our body. Hence the prostaglandins derived from omega 3 fatty acids are known as the good eicosanoids. The bad eicosanoids promote blood clot by platelet aggregation. They cause constriction of blood vessel, promote pain, promote cell division, depress immune system, and depress brain function. So, all the bad eicosanoids are derived from the omega 6 fatty acids. So now this is yet another example. So arachidonic acid is a omega 6 fatty acid. It has 20 carbon chains with 5 double bonds, and it undergoes the Cox and Lox pathway, and it gives rise to series 2 and series 4. The series 2 are prostaglandins, whereas the series 4 are leukotrienes, and all the series 2 and 4 prostaglandins and leukotrienes are pro-inflammatory except PGI2. The icosapentaenoic acid is a 20 carbon PUFA omega 3 fatty acid. When it undergoes Cox and Lox pathway, it gives rise to series 3 prostaglandins and series 5 leukotrienes, and they are all anti-inflammatory. So these are the effects of different prostaglandins and leukotrienes based on the PUFA from which they are generated. Now let us look at the inhibitors of COX. So arachidonic acid gives rise to prostaglandins by the action of cyclooxygenase 1 and 2. So there are two types of cyclooxygenase 1 and 2. The cyclooxygenase 1 is a housekeeping gene and it is always active, whereas COX2 is an inducible gene. So it is activated in different situations when required. Now both these enzymes are commonly inhibited by non-selective NSAIDs, which include aspirin, ibuprofen, diclofenac, and naproxen as some examples. Now the selective inhibitor for COX2 is known as the COX-2 selective NSAIDs, also known as COXIBs. The examples are celecoxib and rofecoxib. So, types of cyclooxygenase enzymes. Cyclooxygenase enzymes are those that act on arachidonic acid to produce different prostaglandins. So, they are of two types, that is COX-1 and COX-2. So the COX-1 is a constitutive enzyme, and which means it is not under regulation and it is always expressed, whereas the COX-2 is an inducible form and it undergoes regulation. So the COX-1 produces prostaglandins and mediates gastric, renal and platelet functions, whereas the COX-2, which is an inducible form, mediates inflammatory responses, and these have only very short life of as much as 30 seconds. So that is how the prostaglandins act by COX-1 and COX-2. So they are also known as local hormones because they have various actions on around the cells that are around them. What is the mechanism of action of aspirin? So aspirin is basically an NSAID which irreversibly acetylates the cyclooxygenase enzyme and inhibits it. So the platelets cannot regenerate cyclooxygenase, and so thromboxane A2 is not formed in platelets, so platelet aggregation can be stopped. So there is decreased platelet aggregation, and therefore a very low dose of 75 milligrams per day aspirin is also used in the prevention of heart attacks. So aspirin is basically a non-steroidal anti-inflammatory drug which is given to patients who are prone to heart attacks at a very low dose because they inhibit platelet aggregation. Now, by inhibiting the cyclooxygenase, aspirin also inhibits prostaglandin I2, which has the opposite function of thromboxane A2. If you remember, thromboxane causes aggregation and PGI2 does the reverse of it. So, this is a temporary effect, so the endothelial cells immediately resynthesize cyclooxygenase again, and so the effect on PGI2 synthesis is not as much. So, aspirin completely blocks thromboxane A2 synthesis, whereas it reversibly inhibits PGI2 synthesis. Thank you. Please like, comment, share, and subscribe to my channel. Thank you for watching.